Hello, my name is Alethea and I attend the University of Connecticut in the Professional Science Masters for Healthcare Genetics program. For this assignment, I chose a very recent publication in Nature Genetics called Proteome Y Model for Human Disease Genetics by Orenbuch et al. This study focuses primarily on missense mutations, which are difficult to detect as the cause of disease in patients because they are very subtle changes and very context dependent. The current models for testing procedures for identifying and categorizing genetic variants, specifically missense variants, are good, but they don't take into consideration a large number of factors, which is why they are limited and why there was a need to develop a more comprehensive model. POP-EVE was developed to solve these limitations, specifically by combining evolutionary and population data. This model is able to find missense mutations and estimate the severity and pathogenicity of the variant. The current, or now considered older, models for identifying causal variants in populations in patients have multiple limitations that include the need to do trio sequencing, which involves doing whole exome sequencing on not only the patient but their parents as well. This is done to narrow down the variants that could be causing a genetic disease, but it still leaves a lot of room for question. Additionally, previous models have a low diagnostic yield, meaning that not a lot of diagnoses are made using these models, which means that there needs to be alternative strategies also used, which uses more resources and time. These older models are able to separate benign from pathogenic variants, but it cannot determine when in the patient's life the affected protein will be disease-causing. For example, disruption of the function of a protein can impact a patient early in their life, or while the loss of function in a protein can have deleterious effects on the patient later in life. So determining whether it's early or late in life is very important. POP-EVE gathers population data from the UK Biobank or Genome Aggregation Database in combination with other computational methods to overcome these limitations without needing to do trio sequencing. So you can sequence the child's exome without needing to do the parental exomes. Some of the methods used in this study include large-scale population datasets such as the ones from UK Biobank and Genome Aggregation Database and multiple cohorts of individuals with moderate or severe developmental disorders. They also developed a probabilistic model using diverse species and human populations to predict variant impact across the proteome, and mathematical models were used to identify conservation patterns across diverse organisms to attain fitness scores that were important for identifying variant impact. So one of the main results that came out of this study came from the combination of evolutionary and population data. The figure shows how data from the protein universe, evolutionary sequences, population variations, and Gaussian processing methods were all combined to see an overall POP-EVE score. The POP-EVE score, unlike other models, are enriched in haploinsufficient genes, as well as in genes with dominant or recessive inheritance patterns, which makes it possible to predict the deleteriousness of a missense mutation in a population. So another significant result that came out of this study was the ability to see variant severity and pathogenicity, which was a major limitation in previous models. As shown in parts A and B of this figure, POP-EVE was able to distinguish between child versus adult death-associated variants as well as predict the age of onset. Interestingly, as seen on the right sides of parts A and B, POP-EVE performs much better and much more accurately than other models such as Alpha Missense and Revel. POP-EVE was also able to identify that variants in patients with severe developmental disorders were more deleterious than those in control cases, as well as able to distinguish between the levels of deleteriousness, which highlights the accuracy and strengths of POP-EVE. Another limitation in previous models has been the overprediction of pathogenicity, which POP-EVE has been shown to eliminate. So POP-EVE is able to separate diagnosed developmental disorder cases based on de novo missense mutations compared to better than other predictors. And it can also recall severe developmental disorder cases based on missense mutations without overpredicting pathogenicity in whole exome sequencing. As with any new technology, the cost and environmental impact are high. Development of large-scale proteomic or genomic models such as POP-EVE is extremely expensive, both financially and in terms of impact on the environment. So for future model development, it is important to take into consideration the computational costs, and researchers should look for ways to recycle and reuse components like they did in this study. One specific issue that was found while developing POP-EVE was, as with any model that uses large-scale population data, is population structure bias. And so in order to address this issue, the developers had a strict measure for missense variation, either marking it as seen or not seen, rather than using allele frequencies like in other models. Due to this fix, POP-EVE shows minimal bias towards European ancestries, and they did some testing to confirm this, while their competitors have been shown to have significant bias towards these populations. Overall, there were many key findings seen in this research article, some of which were not discussed throughout the presentation. With the development of POP-EVE, the researchers were able to identify 123 novel genes that are involved in developmental disorders, and while this is a start, they were able to also identify that there are a lot of other genes that need to be discovered in this field, and more research in this field will help identify those genes. Some of the other findings were diagnosis without parental genomes, so you don't have to do trio sequencing anymore. Also, ranking the variants across the whole proteome, which is a new thing with this technology, because before you couldn't really rank them, you just knew that they existed. Additionally, minimal ancestry bias and capturing the severity of the variant. So overall, POP-EVE has been able to solve many of the limitations of previous models, which creates a more accurate diagnostic method that will hopefully help many patients get ahead of their genetic diseases earlier rather than later.